Team Immunologic Detection of Microorganisms. Plan antigens of bacteria, viruses, and human antigens, autoantigens and heteroantigens, the structure of immunoglobulins, classes of immunoglobulins, interaction between antigen and antibody, the types of serological tests, agglutination test, precipitation test, neutralization test, complement fixation test, and the ELISA enzyme-linked immunosorbent assay, and the PCR polymerase chain reaction. Immunologic detection of microorganisms. A range of immunological tests is used to help identify various kinds of microorganisms. Immunological tests come in many formats, but all rely on the ability of an antigen to react with an antibody. Antigen is a substances, usually protein in nature and sometimes polysaccharides, that generates a specific immune response and induce the formation of specific antibody or specially sensitized T cells or both. Antigen properties. Immunogenicity is determined by foreignness. An antigen must be foreign substance to the animal to elicit an immune response. Chemical nature and composition. In general, the more complex the substance is chemically, the more immunogenic it will be. Antigens are mainly proteins and some are polysaccharides. Physical forms. In general, particulate antigens are more immunogenic than soluble ones. Denaturated antigens are more immunogenic than the native form. Antigen specificity depends on the specific active sites on the antigenic molecules. Antigenic determinants or epitopes are the regions of the antigen which specifically binds with the antibody molecule. Species specificity. Tissues of all individuals in a particular species possess species-specific antigen. Human blood proteins can be differentiated from animal protein by specific antigen-antibody reaction. Organ specificity. Specific antigens are confined to particular organ or tissue. Certain proteins, so brain, kidney, thyroglobulin, and lens protein of the one species share specificity with that of another species. Auto-specificity. Self-antigens are not immunogenic, but under certain circumstances, lens protein, thyroglobulin, and other may act as autoantigens. Types of antigen: heterophile antigens, isophile antigens, and the sequestered antigens and superantigens. Blood type compatibility. The ABO system contains 4 blood groups. The blood groups is determined by the presence or absence of 2 distinct antigens A and B on the surface of the erythrocytes. Distribution of ABO antigens on red blood cells and antibodies in the serum. The donor and recipient should belong to the same ABO group. It used to be held that O group cells could be transfused to recipients of any group as they possessed neither A nor B antigen. O group was designated as the universal donor. The anti-A and anti-B antibodies in the transfused O blood don't cause any damage to red cells of A, B, and AB group recipients, because they will be rendered ineffective by dilution in the recipient's plasma. But some O group plasma may contain isoantibodies in high titers, so that the damage to the recipient cell may result. This is known as dangerous O group transfusion of large quantities of O group blood to persons of another group may also cause reactions. Anti-A antibody in O group blood is generally more potent than anti-B. O group is more likely to cause reaction when given to group A recipient than to those of B group, while O group blood may be transfused to a patient of any other group in the dire emergency. This practice should never be employed as a routine. Due to absence of ISO antibodies in plasma, AB group persons were designated in universal recipients. AB group donors may not always be available due to their rarity, and it may on occasion be necessary to use donors of other groups. In such cases, group A blood is safer than B because anti-A antibody is usually more potent than anti-B. Bacterial antigens, K antigen present in capsule, and H antigen in flagella, and O antigen is somatic antigen. Capsule antigen are divided M antigen, mucous antigen, L antigen, and V antigen, B antigen. Virus antigens, S antigen in nucleocapsid and V antigen on the surface of virus, and H antigen, hemagglutinin and neuraminidase and supercapsid. Antibodies are immune system related proteins called immunoglobulins. Each antibody consists of 4 polypeptides, 2 heavy chains and 2 light chains joined to form a Y-shaped molecule. Antibodies or immunoglobulins are gamma globulins which are produced in response to antigenic stimulation. These react specifically with the antigens which stimulated their production. The immunoglobulins have a basic unit of 2 light chains and 2 heavy chains in a light-heavy light arrangement. There are 5 classes. This antibody structure. 2 H chains are always identical in a given molecule, and the same is true of L chains. Each H chain of immunoglobulin G contains 440 amino acids, while each L chain contains 220 amino acids. H chain has 4 domains of 110 amino acids each, while L chain has 2, and the antigen combining sites of the molecule are at its amino terminal end, and these are composed of both H and L chains. Of the 220 amino acids, those that constitute carboxy terminal half of L chain occur in a constant sequence. This part of the chain is called constant region. Only 2 sequence patterns are seen in constant region of kappa and lambda chains. On the other hand, amino acids sequence in amino terminal half of the L chain is highly variable. The variability determines the immunological specificity of the antibody molecule. It is therefore called variable region. And antibody classes. There are 5 classes of antibody: immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin D, and immunoglobulin E. Immunoglobulin G protects the body fluids. Immunoglobulin A protects the body surface. Immunoglobulin M protects the bloodstream. Immunoglobulin E mediates type 1 hypersensitivity. Immunoglobulin D rule not known. And antibodies neutralize antigens, induce killing of target cell by complement and neutral killer cells, and opsonize particles for phagocytosis. Immunoglobulin G is the most abundant class of immunoglobulins in the body, constituting approximately 75% of total immunoglobulins. Joining chain is present in immunoglobulin M and secretory immunoglobulin A. Secretory immunoglobulin A is relatively resistant to digestive enzymes, which may be due to the secretory components of this immunoglobulin. In contrast to other immunoglobulins, immunoglobulin E is heat labile and gets inactivated by heating at 56 degrees Celsius for 30 minutes. Agglutination reaction: when agents are cell, bacteria, foreign erythrocytes, other cells, agglutination occurs. Agglutination is visible cell congestion, glying. And agglutination reaction: when a particulate or insoluble antigen is mixed with its antibody in the presence of erythrocytes at a suitable temperature and pH, the particles are clumped or agglutinated. Advantages: agglutination is more sensitive than precipitation test, and the clumps are better visualized and interpreted than bands or flocculus, and hence agglutination tests are widely used even in today's modern area of diagnosis. Applications: agglutination reactions are classified as direct, indirect. Indirect is passive, and reverse passive agglutination reaction. All these agglutination tests are performed either on a slide or in tube or in cart or sometimes in microtiter plates. And agglutination reaction, slide agglutination. It is usually performed to confirm the identification and serotyping of bacterial colonies grown in culture. It is also the method used for blood grouping and cross-matching. Bacterial colony is mixed with a drop of saline on a slide to form a uniform, smooth, likely white suspension. Next step is to this drop of the serum containing appropriate antibody is added, and the slide is shaken thoroughly for few seconds. Next process a positive result is indicated by visible clumping with clearing of the suspension, and or if the milky white suspension remains unchanged, indicates a negative result. Latex agglutination. Latex agglutination is in latex agglutination procedures are antigen molecules can be bound to the surface of latex beads. If antibody is present in the test specimen, the antigen will combine with the antibody and form visible aggregates. Hemagglutination test. It refers to the agglutination tests that use RBCs as source of antigen. Various types of hemagglutination tests include direct hemagglutination test. Serum antibodies directly agglutinate with surface antigens of RBCs to produce a MET. Examples include it employs sheep RBCs as antigens to detect Epstein-Barr virus antibodies in serum. The test is performed in tubes. Cold agglutination test it uses human RBCs as antigen to detect mycoplasma antibodies in serum. Test is performed in tubes. And blood grouping, ABO, and rhesus grouping. And the Coombs test or antiglobulin test. It is performed to diagnose rhesus incompatibility by detecting rhesus antibody from mothers and babies serum. Immunofluorescence test is it is a technique similar to ELISA but differs by some important features. Fluorescent dye is used instead of enzyme for labeling of antibody. It detects cell surface antigens. It is also used to detect antibodies bound to cell surface antigens, unlike ELISA, which detects free antigen or antibody. Principle: fluorescence refers to absorbing high energy, shorter wavelengths ultraviolet light rays by the fluorescent compounds and in turn emitting visible light rays with a low energy, longer wavelengths. The fluorescent dye is used to conjugate the antibody, and such labeled antibody can be used to detect the antigens or antigen-antibody complex on the cell surface. The fluorescent compounds commonly used fluorescein isocyanatic and types direct immunofluorescent assay and indirect immunofluorescent assay. Direct immunofluorescence assay. Step 1. Sample containing cell carrying surface antigens is smeared on a slide. Step 2. Primary antibodies specific to the antigen target with fluorescent dye is added. Step 3. Slide is washed to remove the unbound antibodies and then weaved under a fluorescence microscope. Indirect immunofluorescence assay. This detects antibodies in sample. Slides smeared with cells carrying known antigens are commercially available. Step 1. Test serum containing primarily antibody is added to the slide. Step 2. Slide is washed to remove the unbound antibodies. A secondary antibody is added. And step 3. Slide is washed and then weaved under a fluorescent microscope. Application. Immunofluorescent assay has various applications, such as detection of antibodies in autoimmune disease and detecting microbial antigens, rabies antigen in corneal smear, and detection of viral antigens in cell lines inoculated with the specimen. And the next is precipitation reaction. When a soluble antigen reacts with its antibody in the presence of optimal temperature, pH, and electrolytes, saline solution, it leads to formation of the antigen-antibody complex in the form of insoluble precipitate band when gel containing medium is used, or insoluble flocculus or precipitate ring when liquid medium is used. Precipitation in liquid medium. Ring test. In a narrow tube, antigen solution is layered over an antiserum. A precipitate ring appears at the junction of two liquids. Examples of ring test are streptococcal grouping by Lancefield technique and Ascolis thermoprecipitation test done for anthrax. And the flocculation test. When a drop of antigen is mixed with a drop of patient serum, then the precipitate format remains suspended as flocculus. This test can be done on a slide or in a tube. Examples of slide flocculation test used for diagnosis of syphilis, and examples of tube flocculation test. Can test used previously for syphilis. And the precipitation in gel. Incorporation of 1% soft agarose gel in precipitation reaction has many advantages over liquid medium. and it results in formation of clearly visible bands instead of flocculus, which can be preserved for a longer time. It can differentiate individual antigens from a mixture as each antigen forms a separate band after reacting with specific antibody. And the complement fixation test. Complement fixation test is positive and negative serum. Complement fixation test detects the antibodies and the patient's serums that are capable of fixing with complements. It was once very popular, now is almost obsolete. Principle: it is a two-step reaction. Before starting the test serum, inactivation 56 degrees Celsius for 30 minutes is done to remove the non-specific inhibitors of complements present in serum. In the first step, antigen soluble or particulate plus test serum plus genia peak complement all added together. And if the test serum positive for antibody complex is formed, complement gets fixed to the complex, so there will be no free complements in the serum. If the test serum is negative for antibody, next there is no antigen antibody complex. Complements are not fixed, hence remain free in the serum. In the second step, a hemolysis indicator system is added. It consists of sheep RBCs coated with its antibodies called amboceptors and prepared by injecting sheep RBCs into rabbit. If the test serum is positive for antibody, there is no free complement in serum to bind to amboceptors. There is no hemolysis. If the test serum is negative for antibody, the free complements are fixed to amboceptors bound of sheep RBCs. Result in hemolysis. Applications. In addition, complement fixation test was also widely used for detection of complement fixing antibodies of rickettsia, chlamydia, brucella, mycoplasma infections, and some viral infections such as arbovirus, rabies, and etc. Indirect complement fixation test was used in the past to detect certain avian and mammalian serum antibodies which cannot fix genia peak complement. And toxin neutralization test. Neutralization tests are also less commonly used in modern dyes. Various examples are as follows: viral neutralization test and toxin neutralization test. A viral neutralization test it detects the presence of neutralizing antibody in patient serum when the serum is mixed with a leaf viral suspension and poured onto a cell line. Specific serum antibody neutralizes the surface antigen, making the virus unable to infect a cell line. Viral neutralization is of two types: irreversible neutralization and stable neutralization. Plaque inhibition test. This is a neutralization test done for bacteriophages. Toxin antitoxin neutralization test. Examples include Schick test and the ASO test and Nagler's reactions. Schick test, it is a diphtheria toxin antitoxin neutralization test. It was used in the past to know the susceptibility of individuals to Corynebacterium diphtheria. And ASO test anti-streptolysin O antibody was detected before by neutralization method. However, it is now replaced by latex agglutination. And the next ELISA test, enzyme linked immunosorbent assay, and direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. ELISA is so named because of its two components, immunosorbent and enzyme. Immunosorbent, here an absorbing materials is used that specifically absorbs the antigen or antibody present in serum. Enzyme is used to label on one of the components of immunoassay, antigen or antibody. Substrate chromogen system. A substrate chromogen system is added at the final step of ELISA. And ELISA is usually performed on a microtetra plate containing 96 wells, or less commonly performed in tubes. The microtetra plate or the tubes are made up of polystyrene, polyvinyl, or polycarbonate material. And the enzyme reacts with the substrate, which in turn activates the chromogen to produce a color. Sometimes the substrate is chromogenic in nature. On reaction with the enzyme, it changes its color. The color change is detected by spectrophotometry in an ELISA reader. Intensity of the color is directly proportional to the amount of detection molecule present in test serum. Types of ELISA. Yes, direct ELISA. It is used for detection of antigen in test serum. Here, the primary antibody targeted against the serum antigen is labeled with the enzyme. Step 1. Wells of microtetra plate are empty, not pre-coated with antigen or antibodies. Step 2. Test serum containing antigen is added into the wells. Antigen becomes attached to the solid phase by passive absorption. Step 3. After washing, the enzyme labeled primary antibodies raised in rabbits are added. Step 4. After washing, a substrate chromogen system is added and color is measured. Indirect ELISA. It is used for detection of antibody or less commonly antigen in serum. It differs from the direct ELISA in that the secondary antibody is labeled with enzyme instead of primary antibody. The secondary antibody is an anti-species antibody, anti-human immunoglobulin, an antibody targeted to Fc region of any human immunoglobulin. Indirect ELISA for antibody detection. Step 1. The solid phase of the wells of microtetra plates are pre-coated with the antigen. Step 2. Test serum containing primary antibody specific to the antigen is added to the wells. Antibody gets attached to the antigen coated on the well. Step 3. After washing, enzyme labeled secondary antibody, anti-human immunoglobulins is added. Step 4. After washing, a substrate chromogen system is added and color is developed. Sandwich ELISA, it detects the antigen in test serum. It is so named because the antigen gets sandwiched between the capture antibody and the detector antibody. There are two types of sandwich ELISA, direct and indirect, depending upon whether the detector antibody is a primary antibody direct or secondary antibody indirect. And polymerase chain reaction. Polymerase chain reaction is a technology in molecular biology used to amplify a single or few copies of a piece of DNA to generate millions of copies of DNA. It was developed by Carl K.B. Mallis in 1983, for which he and Michael Smith were awarded the Nobel Prize. Principle polymerase chain reaction involves three basic steps: DNA extraction from the organism. This involves lysis of the organisms and the release of the DNA, which may be done by various methods such as boiling, adding enzymes such as lysozyme, proteinase K, and etc., phenol, chloroform, and isoamyl alcohol methods. DNA extraction kits are also available commercially. Second is amplification of extracted DNA. This is carried out in a special PCR machine called thermocycler. The extracted DNA is subjected to repeated cycles from 30 to 35 numbers of amplification, which takes about 4 hours. Each amplification cycle has 3 steps. Denaturation at 95 degrees Celsius. This involves separation of the dsDNA into 2 separate single strands, and the second step is primer annealing. 55 degrees Celsius primer is a short oligonucleotide complementary to a small sequence of the target DNA. It anneals to the complementary site of the target ssDNA, and the last step is extension of the primer 72 degrees Celsius. This step is catalyzed by Taq polymerase enzyme, which keeps on adding the free nucleotides to the growing end of the primer. And the last step is gel electrophoresis of amplified product. The amplified DNA is electrophoretically migrated according to their molecular size by performing agarose gel electrophoresis. The amplified DNA forms clear bands which can be visualized under ultraviolet light. Amplification of PCR. Polymerase chain reaction is now a common and often indispensable technique used in medicine, diagnostics, and research laboratories for a variety of applications. It has the following advantages compared to the conventional culture methods. More sensitive, it can amplify very few copies of the specific DNA, so it is more sensitive. More specific use of primers targeting specific DNA sequence of the organism makes the PCR assays highly specific. PCR can be done to amplify the DNA of the organism, either directly from the sample or to confirm the organism grown in culture. PCR can also detect the organisms that are highly fastidious or non-cultivable by conventional culture methods. PCR can be used to detect the genes in the organism, responsible for drug resistance, detects genetic disease such as sickle cell anemia and phenylketonuria and muscular dystrophy.